Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. And ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 95th seminar. And I call it on LBNL, or Lawrence Berkeley National Laboratory Day. Today is a very special day to me. Today is my mother's birthday. So happy birthday to my mom. And regardless of my age, I may be still a child to her. Regardless of what I'm doing, good or bad, all the moms loves, of course, their children. I want to say I love you, mom. And this one saying may be more than sufficient or better than any other gift. I also want to say all moms are Wonder Woman and all working moms are super Wonder Woman. And thank you, all moms. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Aindrila Mukhopadhyay. She is a science deputy for the bioscience areas, biological systems and engineering, and also senior scientist at Lawrence Berkeley National Lab, and also a vice president of biofuels and bioproducts at JBA, where Jay Kiesling, another pioneer speaker who gave brilliant talk 2 years ago, I believe, is our CEO. I'm also so glad to see her again because my recent in-person meeting was very short in Minnesota, and I could not meet her when I visited UC Berkeley last month. Aindrila, thank you so much for your great contribution to the scientific society, including the great conference that you chaired in Minnesota this year, and the virtual podium is all yours now. Thank you again.
Thank you, Tae-Suk. That's a very kind introduction. I will share my screen. I have a few slides.
Wonderful.
All right. So I'm gonna just use a little bit of you all's time to tell you a little bit about where I'm from and the work that my team does. So we primarily focus on discovering and engineering new functions in microbes. I think that kind of captures what we do. So as Tae-Suk said, I'm from the Lawrence Berkeley National Lab. Lawrence Berkeley National Lab main campus is up on Berkeley Hills. It is the most spectacular place for research and also for the views. And right uphill from the UC Berkeley campus, that's where we get our logo from. That's the light source and the clock tower of Berkeley campus. And so we are primarily Department of Energy funded lab, and our work is very team science. So why I'm at this lab is because when I joined this lab many years ago as a postdoc, I was exposed to what team science can do and how much further my efforts can do if I work together with other people from other disciplines. And I think this was the foundation on which Berkeley Lab was based, and it really works for certain kinds of mission-driven research, and that is why I remain at Berkeley Lab. As a result of this, I am part of many very large team projects, and I'm showing you a few of them here. I'm part of the DOE scientific focus area, Enigma, which primarily studies microbes in DOE-relevant environments. I'm part of MCAFEs that also studies SynComs and other communities in the plant root system to understand how these co-benefit each other and how we can use them for important targets of carbon sequestrations. And I'm also part of the Joint Bioenergy Institute. And I'll tell you a little bit more about JBay. So JBay is a bioenergy research center. As Taeseok said, it's led by Dr. Jay Kiesling. It has partners from all over the country and now also from Australia with Jenny Mortimer. And the mission of JBay is to establish the scientific knowledge and new technologies to transform the maximum amount of carbon available in bioenergy crops to biofuels and bioproducts. And so a little bit of a background. You know, there are a lot of things that come out of a barrel of oil. And this is the barrel of oil, the petrochemically derived, non-sustainable products that we are aiming to replace. Much of it is fuels, but there are also many other components and products in our lives that are coming from unsustainable or non-sustainable carbon sources. And in fact, there is an opportunity here to build the bioeconomy that can address many aspects of this challenge, including replacing fuels, reducing greenhouse gas emissions, and creating jobs. So that's sort of the overarching goal under which the Bioenergy Research Centers sit. And microbial conversion can play a very critical role in this goal. So here we have various carbon sources that are sustainable. One amongst them is plants and crop and energy plants that can serve this sustainable carbon source that we want to convert into a range of compounds. So just to give you a visual on what kind of compounds we want to convert, not just sugars that are commonly used in biotechnology, but also a wider range of sustainable carbon sources into biofuels and bioproducts. Biofuels tend to be hydrocarbon compounds and compounds with these ring structures that are high energy, that can be used all the way from replacements to automobiles, more importantly, for aviation fuel and other high energy requirements in long-haul trucks, etc. Whereas bioproducts tend to be more heteroatom-containing compounds because they form precursors to polymers, plastics, commodity chemicals, platform chemicals that have a much wider range of applications. So I wanted to give you one example of a bioproduct. This compound is called indigoidine. So indigoidine could be an alternative to indigo, which is a blue dye. As you all know, you all wear blue jeans. Many of you are probably wearing a blue jean right now. Blue jeans come at a cost to both the environment and to society. It uses a lot of water. It results in a lot of CO2 emissions, and it uses a lot of chemicals that are not necessarily benign to our environment. And one of them is the production of the dye and the use of the dye. So here we are trying to figure out ways to make these kinds of chemicals in a sustainable manner. So what we are trying to do is to use microbes to convert a range of carbon sources to produce these kinds of molecules. And this we can do very, very well. Indigoidine is actually a really simple molecule to make. It takes 2 genes to engineer any number of organisms to make it. But the point is, how do we make it in a manner that makes it a production host for scaling? This is a much harder challenge because many different aspects of the microbe needs to be engineered if you want to make it a scalable host. It has to consume all the biomass chemicals. You have to be able to engineer it. It has to tolerate hydrolysate components. It has to have very high flux to key intermediates or products. It has to maintain the right metabolism when you scale it up. And it has to tolerate any intermediates or final products that are inhibitory to it. And this is goes beyond pathway engineering into strain and metabolic engineering. So in our team, we are trying to figure out how we go from what we imagine microbial cultures are, these very simple broth-like cultures, to these kind of cultures, which is what the reality of scale-up from sustainable carbon sources looks like. So in order to achieve this, we've done a large number of very integrated ensemble methods. Don't have the time to tell you about that, but if you would like to see a recent example of our work, a very nice set of work from a big team of scientists at Berkeley Lab and beyond just came out in Cell Reports. This is led by Thomas Eng and Dipanvita Banerjee in my team, where they used a range of methods to iteratively improve the production of indigoidine from aromatic compounds, reaching over 70% max theoretical yield from aromatics in a single study. Of course, there's a ways to go. We have to figure out how to use mixed carbon sources. We have to implement these approaches to many final products, and we have to lower the process requirements to make these processes techno-economically viable. So that's sort of like the essence of the work that we do in our group. I did want to give thanks to the actual people in the group that do the work. They're a really, really cool bunch. And I wanted to thank the funding agencies for the funding we get for the MCAFE's Enigma and JB project. All of them are funded by DOE BER. I also have funds from Berkeley Lab. I'm part of projects that are funded by NSF, DARPA, and I also get support from campus. So I wanted to take this opportunity to tell you that we have 2 open postdoc positions in my team, one in a microbial environmental microbiology project, and one in a metabolic engineering project. So if you're interested in them, check us out and get in touch with me, and I'd be happy to tell you more about that. And with that, I'll turn it back to Taeseok.
Fantastic, fantastic. You know, I want to say one more time, why not? This is great advisor and great environment, great team members you could work with. So apply, please. I want to apply myself. So that's amazing. So thank you so much. It's amazing. I 100% agree with you. Collaboration will make things happen. And then without collaboration, we cannot solve all the problems. So thank you so much emphasizing that important aspect of research. And thank you. Amazing, amazing, Tom. Thank you. All right, so now the main speaker of today with a longer introduction, Dr. Michaelle Mayalu is an assistant professor of mechanical engineering at Stanford University. She received her PhD, MS, and bachelor degree in mechanical engineering at MIT. She was a postdoctoral scholar at the Caltech, and she was a 2017 California Alliance Postdoctoral Fellowship Program recipient, and also a 2019 Borough Wellcome Fund Postdoctoral Enrichment Program Award recipient. Her area of this expertise is in mathematical modeling and control theory of synthetic biological and biomedical systems. She is interested in the development of control theoretical tools for understanding, controlling, and predicting biological function at the molecular, cellular, and organismal levels to optimize therapeutic intervention. Her research objective is to investigate how to optimize biomedical therapeutic designs using theoretical and computational approaches coupled with experiments. Initial project concepts include, first, theoretical and experimental design of bacterial micro-robots for therapeutic intervention. Second, system-level multi-scale modeling of gut-associated skin disorders for virtual evaluation and optimization of therapy. Third, theoretical and experimental design of micro-robotic swarms of engineered bacteria to explore possible mechanism of pattern formation. Ultimately, her lab aims to develop accurate and efficient modeling frameworks that incorporate computation, dynamical systems, and control theory that will become more widespread and infactable in the design of electromechanical and biological therapeutic machine. I believe she's a true rising star with a huge potential, and I wanted to thank her again for your time today. And please and take it away. And thank you so much again.
Yeah, thank you so much, Dr. Moon. Thank you so much for the introduction. Let me go ahead and share my screen very quickly. And presentation mode here. All right.
Wonderful.
Great. Yeah, so I think Dr. Moon did a great job in basically introducing me. So thank you so much. In the department here of mechanical engineering, I just got a bioengineering courtesy appointment too last month. So something I want to mention as well. But today I'm going to be talking about the analysis and design of engineered cell population control circuit with a specific control architecture called paradoxical feedback. So first, I'd just like to introduce a little bit more about the research agenda in my lab. So our research focuses on the treatment of the human body as a collection of control system modules at multiple levels. So ultimately, we would like to use principles from electrical and mechanical system design to understand and eventually rebuild biological systems. So here I'm just showing a wide range of biological phenomena at different scales. And all of them have some type of control or feedback control that involve them. So here I'm showing kind of examples at the multi-organ levels of control include locomotion, active sensing, vision, proprioception. At the single organ level, we can think about cardiovascular feedback mechanisms. At the multi-cell level, we could think about control in the metabolic and hormonal and immunological control mechanisms and how they communicate through signals. And then at the single cell level, we can think of gene regulation and signal transduction and how those, potentially can connect and be described in a control system way. So what I've mentioned are kind of the natural control systems that are inherent in the dynamics of the human body. But there are multiple opportunities for manipulation of these control systems through external inputs. And this can include things like antibiotics or potentially diet. But here we're going to focus on the introduction of engineered cells and how they can act as smart therapeutic devices to regulate function. And also by studying biological systems, again, I am in the mechanical engineering department here. So I'm studying biological systems from a mechanical engineering viewpoint. So hopefully we might start to see things in biological systems that might not have been captured from maybe a biological viewpoint, or might not have been seen or understood before. So from this new viewpoint, hopefully will come new language, new methodologies, new agendas, and hopefully these will all be important and can contribute to mechanical, electrical, and bioengineering fields. So just as a recap, the definition of a dynamical system is a system whose behavior changes over time. So a lot of things fall under this category, including cars and aircraft. So these are more kind of things that we might think about when we think of dynamics, but also the human body is a dynamical system, as are cells. And the way we can regulate the behavior of dynamical systems is by implementing a control system. So here I'm showing a closed loop or feedback control system. And here the output of the control system or the process, is being fed back to regulate the behavior of the system and increase the accuracy of control. So in control theory, we actually develop a mathematical model of our system or our process, and then we derive a controller or some kind of control input that we can use to drive the system to a desired state with optimal stability while minimizing things like delay, overshoot, or steady state error. So parts of a control system, some main parts of the control system are the actual controller. So this is where you compare your behavior, your output behavior, to your actual desired behavior. And based on that, the controller sends a signal to an actuator or an effector. And this actuator is what makes adjustments to the actual process, the dynamics of the process. So once the process changes, we have a sensor that senses that change, and that change is then fed back to the controller and back until we've driven the output to the desired behavior. And so some notable applications, some traditional applications of control theory are cruise control for a car, and also aft control for the aircraft. So both these systems have multiple layers of equipment to provide continuous, reliable feedback and control of the airplane. But we can also think about how feedback control is pervasive in biological systems. And one example is calcium homeostasis in mammals. So here, different organs in the body work to maintain the calcium concentration in the blood to a specific set point. Another example is bacterial chemotaxis. So this is where the tumbling rate of bacteria can be maintained at a constant level. So one big difference in biological control systems is that the separation of sensing, actuation, and computation, as I kind of showed here, it's less evident. So instead the dynamics of the molecules that sense the environmental condition and make changes to the operation of the internal components, they are kind of jumbled together. They're all integrated together. And it's not that easy sometimes to untangle. And so the idea of kind of having this set point or a reference value, sometimes it may not be an explicit signal, but it could be a consequence of many different changes or parameters or regulatory elements within the system. So engineers have also fabricated architectures at the genetic and multi-cell levels. So genetic engineering is one of the kind of seminal kind of control architectures is the genetic toggle switch. So this is where we can use different concentrations of chemicals to toggle a gene on or off. We can also think on the kind of multi-cell scale of genetic engineering. This is also a pretty seminal paper, which looks at coordinated communication through engineered cell-cell signaling mechanisms. And this is to maintain the cell population at a constant level. So these control systems, they must also manage uncertainty in the biomolecular process to be controlled. They have to manage environmental disturbances, and they also are context dependent in many cases. So using control theory, we're able to generate hypotheses to direct conceptual understanding and extract the underlying fundamental principles of these biological processes, specifically in therapeutic contexts. So I'm going to talk about one of the main research areas in my lab, and that is, again, to create these kind of genetically engineered or microrobotic therapeutic systems. And these are using cells, bacteria, and they are driven by genetic components or biomolecular components. So hopefully these systems, they'll be able to kind of quickly adapt and correct disease states when input into the body. So our agenda then is to construct this diagnostic, therapeutic, and treatment devices, and using the same materials that make up your body. So these are cells and microbes. And we also want to design biological control modules specifically that are safe, reliable, and controllable. So these are the main kind of constraints or ideas that kind of drive our design. Biosafety and robustness. So things like that I will talk about or later are things like emergency shutdown. So we're able to kind of shut down this system in all situations. Safety, so maybe some systems that are more robust to mutations and also rejections of disturbances. So in order to build these types of devices and perform our analysis, we need to draw upon many different subjects. So control theory is one, but also, we might be looking at machine learning or computation. Nonlinear dynamics is a big part of this. Bioengineering, obviously, medicine. We want to be able to kind of incorporate all these different subjects and synthesize them into kind of treat or help with our specific agenda. Okay, so let me get into the specifics of the project I will be talking about today. And so it has to do with detection and treatment of infections. Specifically, we want to build an engineered bacteria that can, when there's an infection in the gut, it can somehow sense the signal of that infection. And based on the signal it senses, it initiates our engineered cell system to expand. So they start off on a small population, and based on the signal they sense, they grow into a larger population. And then they start treating the bacteria. We also want to make sure that we have safety in mind. So in any point during this process, we have the kind of doctor administered signal, or this remote signal, that can attenuate or shut down these program microbes, basically eliminate them from the body at any time. If we're not using this shutdown switch, we basically initiate the program microbes to grow to a certain population. Once the infection is kind of gone, and the program microbes no longer sense the signal, the microbes will collapse back to their smaller density and stay there. So the main design specifications for this system, we want to be able to basically treat a pathogen within the gut environment. We want to have robust population control. As you see, we have this idea of expanding and contracting between two population levels for kind of this robust bistable switching. And we also, with this type of bistable switching, we want to have this kind of automatic and preemptive detection of the pathogen, possibility of kind of repeatable treatments, and the idea of this emergency shutdown. So these are kind of the specifications that we would like for our system. So let's talk about some previous works that we could draw some inspiration from. So there have been previous works from genetically engineered E. coli that can detect pathogens like Pseudomonas by sensing signals that Pseudomonas releases and responding by producing a specific toxin that's specific to that specific pathogen. These are some of the previous works that have done that. One main thing that hasn't been addressed within these previous works is potentially population control. So one can see if this thing is going into the body and these bacteria are growing at larger populations potentially once they, if they sense a signal, they're growing without balance, without control, there could be kind of a containment issue that could occur. We want to kind of add that safety mechanism back into this type of design. Another thing that we've seen that are not specific to this gut treatment idea, but that have been done is just robust population control in general. So what I'm showing here, this was in collaboration while I was a postdoc at in Caltech. I was in collaboration with the Elowitz lab. We looked at kind of how robust population controllers, this specific one is called paradoxical feedback control, how it could potentially be more robust to mutations. So in the case where we had a more simple control, population control, we had that mutations at some point, we had a mutation that allowed the controller to stop working and the cells would just grow, without bound. But in the case where we had our specific paradoxical feedback, which I will go into in detail. So this feedback is basically a little bit more, in detail in terms of the architecture. We were able to see that the mutations, even if there was a mutation to occur, we would still have the cells maintained below the carrying capacity. So the controller could be maintained at a specific set point for a longer duration or the duration of the experiment. So here again, this was kind of a proof of concept that we did. So it wasn't in the context of a synthetic biology probiotic. And this type of controller architecture hasn't been done in E. coli. So our work, we would like to analyze the programmable capabilities of these kind of robust population controller architecture, specifically this paradoxical feedback architecture. We want to analyze it in the context of pathogen detection within the gut environment. And we want to, within this architecture, ensure that we're able to have this kind of ability for emergency shutdown. We want to ensure kind of safety and robustness, and we want to ensure we have some kind of method for disturbance rejection. Okay, so now let me explain a little bit more about the differences between different feedback controller designs for population control. So the first thing I'm showing here is the negative feedback control. So this is where you basically have a cell population that secretes a small molecule inducer. Here it's secreting a quorum sensing molecule. And this quorum sensing molecule is interfaced with a cell death gene. So as the population increases, we have the concentration of the quorum sensing gets higher and higher, and the cells will, kind of at some point maintain a level below at which they can actually grow because they're connected to that cell death gene. So if we were to show how this looked in terms of the net growth rate versus the feedback signal, what would look like would be this monotonically decreasing curve. And there would be a point here. This is the point of homeostasis, where you, if you're above this point, you're in a state of negative net growth. And if you're below this point, you're in a state of positive net growth. So you can see that this is a kind of a stable population that once the cells grow up to this point, they kind of stay stable at this point. If they grow a little bit, they will die until they reach this point. If they die a little bit, they will grow until they reach this point. So this is called a monostable population dynamics. So in paradoxical feedback, we have both positive and negative feedback. So this is where we have, again, this quorum sensing molecule that is not only interfaced with a cell death gene, but it's also interfaced with a cell growth gene. So we have both positive and negative feedback that are occurring. And in different, if you kind of tune your positive and negative feedback correctly, you can get this type of dynamics. So you can get dynamics that are kind of this biphasic curve. And here we have 3 points here, kind of these 3 equilibrium points. And it turns out that we have 2 of them that are stable, and then we have an unstable middle point. So if we have the quorum sensing signal that is, or feedback signal that's within this area, we have the cell population will be driven or attracted to the low steady state point. And if we have the cell population, it's in a higher area, in this area, the signal is in this higher area, the cell population will be driven to this higher population level. So we kind of have these 2 bistable population levels, and this is called the bistable population dynamics. So the question now is, how does this help with robustness? So let's think of a scenario where we have a mutation, a specific type of mutation. So in this mutation, we're no longer able to sense our quorum sensing molecule. And in the case where our mutant cells aren't able to sense their environment or sense their quorum sensing secreted molecules, or they sense it very, very small, we're basically stuck. The cells are stuck in this constant state of growth. They never get the signal to die. So because they're stuck in this constant state of growth, in this mutation, those cells will keep growing and kind of take over the control. So, you will not be able to maintain the population below a certain level. In the case where we have paradoxical feedback, in the same scenario where we no longer can sense any cells, any quorum sensing, we can't sense the environment, we're still in a kind of a level of low or no sensing, but instead of being in a state of positive growth, we're in a state of negative growth. So instead of growing, these cells will die. And they'll die and they'll reach this low population level, and we will maintain control. Basically, we will not have this aberrant overgrowth of cells. So that's why we care. So that's why we see that this type of architecture is robust against these types of sensing mutations. So that's one aspect that we care about. The other aspect, so this was work done by my graduate student Yu Lin. He basically kind of took advantage of this inherent bistability of the paradoxical feedback architecture to try to see if we could control the switch between our lower level steady state and our higher level steady state using an external signal. So basically, you have some external homologous chemical that potentially will add to the environmental amount. And once that external signal is felt by or sensed by the cell population, it triggers them to be shifted into a state of positive growth, net growth, and then they will just proliferate until they reach the higher level steady state. And if you're able to remove that external homologous signal, this is reversible. You can flip back to being in the state of negative growth and then be attracted to the lower level steady state. So that's the idea, to kind of create this population level switch, use an external signal to potentially go from a low level to high level. And in the context of the infection, basically gut infection detection, what we would do is we would have the infection here. So this is potentially a Pseudomonas infection. Just like before, the Pseudomonas, it kind of releases a signal. It actually releases a quorum sensing signal. And if you design the controller, we basically design the controller in such a way that the feedback that the engineered cells are using is also using a homologous quorum sensing signal to the infection, such that when the infection is present, it acts like it's an added signal to the architecture, and if it's in a larger amount, it can be shift basically your population of engineered cells from that low level to the high level, like I showed on the previous slide. So that's the idea of the architecture here. So the main things are, again, our activation signal, which is the infection signal itself, which kind of is what changes the engineered cells from their deactivated state, which is the low level population, to their activated state, which is a high level population. And then we've incorporated these emergency shutdown. So this is where we've had the idea to have kind of a signal that represses the production of the transcription factor necessary to basically for the controller to work, to turn on the controller. So this transcription factor is basically what is necessary for the quorum sensing molecules to bind with, and then interface with the growth and death genes within the system, and implement the paradoxical control. So if that transcription factor is not present, there's no way for the controller to turn on, and the cells will just die off. They'll be dead. So that is that's the second factor. The third, excuse me. So yeah, the third factor we have here is the actual controller itself. So what we've done first for proof of concept for the kind of growth mechanism or the growth feedback mechanism, we want to interface our quorum sensing molecule with a drug resistance gene. And that in the drug resistance gene is basically acting against some drug which we have constantly within the environment. So if the drug resistance gene is off, we have that the drug will kill the engineered cells. If the drug resistance gene is on, we have the cells will persist and grow. And then we also have a toxin. This is the death part of the circuit. And so this is a toxin that if above a certain threshold, the engineered cells will die. So we have basically growth by drug resistance and we have death by the toxin. And then we have the emergency shutdown where if these two arms are off, the environmental drugs will kill everything. Hopefully that's a little bit clear. And then lastly, we have the actual production of our quorum sensing or feedback signal. So here again, it's a quorum sensing molecule, an AHL that's homologous to our activation or infection signal. So those are the main portions of the design. And so using mass action kinetics, we can actually write down the equations that kind of represent the components within the system. And we also have to couple these biomolecular components to the actual cell level dynamics. So we use both mass action kinetics and we couple it to logistic growth dynamics here. We also use kind of time scale separation. Since we know something about the parameters within the system, we can separate the time scales to reduce the model. And we also use stability analysis to understand what type of parameter regimes will allow us to be in this kind of bistable growth dynamics. So that is how the model briefly was made. So based on the model now, we can try to understand, different simulations and, try to analyze the system using different simulations. So here we're just verifying that the system is in fact bistable for the parameters we've chosen. So we can see here that even, with the addition, of our feedback signal and our activation signal, we still have this kind of bistable, we're still able to maintain this bistable architecture. And then we were wanting to understand basically the balance between different parameters, specifically the gene expression of our negative and positive feedback. So we varied the gene expression of the toxin and drug resistance. And we saw that, depending on the degree of gene expression for both of each of these genes, we could get different stability characteristics. So there's really a balance between the gene expressions that allows you to get this type of bistable net growth versus, quorum sensing architecture. So that was something that we had to, make sure that we were in the correct regime. And then we basically wanted to verify that we were actually able to kind of detect the infection and that the system would, actually act as necessary. So here, what we showing is that before there's any infection, starting at an initial cell population that's below our unstable equilibrium point, we are driven to the low stable steady state. Then when the infection signal is introduced, from the low stable steady state, we are basically can jump to the area that's above this low equilibrium point and go to the higher equilibrium point. So we're driven to this higher equilibrium point. So one thing you might notice, or I should mention, is that because we're changing the parameters of the system, the actual equilibrium points shift. So we have, one set of equilibrium points here. We've changed the parameters, and we've gone from 0 infection to a non-zero infection, the dynamics switch, but we're still able to get to that high steady state, that high steady level. Now, when we remove the infection, our initial point or initial population state starts off, at where we were previously. But because we have removed the infection, basically the infection is now back at 0. Our dynamics have now switched back to the original spot. And it's in such a way that the equilibrium point, basically our initial point is below the unstable equilibrium point, and we're driven back to the stable point here. So yeah, that's kind of the kind of ins and outs of some of the dynamic analysis that we did. We also, again, verified for mutational robustness. We saw that kind of this is as the safety. When we had that loss of sensing mutations, we were also able to recover. So that we also did that simulation as well. And then the shutdown. So this is where, again, I'm just showing more of the equations that we had to represent the basically how we had the off signal repress the production of the transcription factor. And basically from those equations, we could simulate how even in the presence of infection, we were able to kind of turn off completely, irreversibly, the system. So we were able to, here I'm showing where there was no infection occurred, and then when the off signal is introduced, we see that the cells just die off. So lastly, I'm going to talk a little bit about some of the current project progress. We want to be able to optimize, different areas within the control. So what we've understood, or what we see from the analysis, is that increasing the transcription factor production increases the detection sensitivity. So we're able to detect, the infection at lower levels, but it also decreases our ability to reject any disturbances. And this is because as we, again, kind of varying the parameters within the expression of transcription factor within the system, we see that increased transcription factor expression changes the equilibrium point, shifts the equilibrium point such that the unstable equilibrium point shifts lower. And this is what is attributed to having a system that is more sensitive. So we're able to basically, it's easier for us to kind of jump from this low steady state into this kind of high steady state attraction area. But also, if we have any disturbances, we have this problem where we might turn on when we don't want to turn on. The population might expand with some environmental disturbances, or we don't want it on. So we want to be able to balance that rejection to disturbances, but also having a detection that's sensitive enough. And so what Yulin has been working on now is how to kind of optimize the threshold to detect infection with the effect of disturbances, using optimization based ideas from control theory. And the idea is to kind of minimize the threshold to detect infection as a function of the transcription factor expression, such that the effect of disturbances is below some specific level. And so that's what he's doing now. He's also looking at kind of a more stochastic way to treat the problem, where he's looking at agent-based stochastic simulations, where you can see now that kind of maybe a more realistic prototype where he has an infection being introduced. He's having the engineered cells treat that infection, and then showing how when introduction of the off signal, basically kills all the engineered cells, regardless of if the infection is there or not. And so this is also ongoing, and we're also going to see if we can analyze this kind of stochastic, this stochastic model as well. In a more theoretical way as well. So yeah, now I'll just give my concluding remarks. So basically what we've done so far is kind of create this population level bistable switch in the context of infection detection within a gut environment. It basically has this topology that has both negative and positive feedback for when we have a single molecule being secreted by the cell. And then we have a homologous signal that can kind of perturb the system such that it can be kind of expand or contract into different states. And then basically the idea behind this topology, we wanted to always keep in mind the idea of biosafety and robustness. There's also a lot of different topologies in nature that have this, this similar paradoxical architecture. So we have this, single, chemical that kind of feeds back either both positive and negative signals. So we want to be able to have some of the some of the analysis or some of the ideas from our design potentially be useful in different cell types, even in that have the same topology and potentially create, cells that can be useful for different areas in healthcare or other areas. So what's next is to, again, I've talked a lot about the detection part, but we also want to incorporate the treatment part. So we want to be able to have a very kind of simple but sophisticated, as well as sophisticated design to as well as detect, but also treat these the Pseudomonas or the bacteria or the infection within the gut. We also want to make sure that we're taking into account burden of the circuit. We want to address potential failure modes of our circuit as well. And obviously, last but definitely not least, is experiments. So my lab has just finished renovating. So we are very excited to start experiments and to see how our theory can be implemented, practically within the lab. And with that, I would like to thank most specifically my group. I'm very grateful to have students, within this year mark, and also some of my early collaborators and my early funders, which include the Burrow Wellcome Fund and the Hypothesis Fund. Also, I'm also hiring grad students and postdocs. And as you can see, we're very interdisciplinary. We do a lot of math and theory. We also do a lot of computation. And we are starting experiments. So if you have any experimental postdocs who are interested in this type of work, or even experimental students, I would love for you to reach out to me. And yes, that's the end of my talk.
Oh, fantastic. Yeah, I love it. Again, apply, why not? This is one of the best lab, rising labs. So apply. So this is fantastic talk. Let me see. I want to start asking one question. I have many questions, but I cannot do so because of time. So the one question is, as you know, I'm very interested in biosafety, kill switch. We published a couple of papers about that, and then you can address some of that. And one thing I always thinking about in terms of metabolic burden or genetic circuit burden, and you mentioned at the end of your talk. Though my question is, I used to believe complex circuit is the best one, but now I'm changing a little bit, because if you add one more genetic part, the caveat is you have the additional burden into the system. So we I'm thinking about, I was thinking about the complex circuit, and then you also thinking about some a little bit complex circuit in the end. But could you kind of comment on that, trade-off between adding complexity and additional burden and
Yeah.
so?
Yeah, great question. I think that's very obvious, and so we also have that idea, and we're also always thinking about our mantra is to try to get the simplest, most kind of elegant design that can do what we want, and so a design that also could be useful for different things. So within the paradoxical architecture, one function of having that architecture is, it can be mutationally robust. So that's one function. The other function is that it's bistable. So without, adding anything to the circuit, we can kind of manipulate it in such a way that it can now act as a detector. So instead of adding anything new, we have a detector and we have a kind of a robustness module. Now, when we kind of incorporate treatment, we want to, again, see if we can try to take advantage of the design, this bistable architecture, and, kind of help it create a simple treatment module that also utilizes this paradoxical framework and architecture. So that's something that my student also is, thinking about and seeing if we can somehow use, the switch-like idea and connect it to the treatment as well.
That's wonderful. So I have the second question because I didn't see any question from audience. My second question is I love your paradoxical circuit architecture. Then also you address the mutational instability. That's amazing because when we develop kill switch, we basically suffer from mutation entire 60 years. So we at the end of the day we overcome by doing of course the redundant circuit. So we have the multiple kill switch. In that way we address the problem.
Yeah.
But at the same time, that require, because we add one more circuit for the kill switch, I mean, for complete killing, that is biocontainment issue. So we need to have the absolute elimination of all the living engineered microbes. So we achieve that one. But for some application, like let's say you use a probiotic, and then let's say probiotic only stay a couple of days in your gut, the mutation not necessarily affect the entire performance if the mutation rate is not higher than whatever 1 or 2 days time frame. So can you kind of comment on some specific application that is not necessarily require very complex circuit? And you mentioned simpler the better. So what is your view or perspective on that point?
Okay, so trying to see if we can create a circuit that may not have to account for mutation because the amount of time it's going to be within your body, mutation is not a problem. Yeah, that's a good point. That's a great point, actually. I feel like the circuit that we have designed now, even if the kind of our robust architecture isn't necessary, I think the idea of it being kind of bistable, where it can expand and contract to different populations, is something that is good, even if your cells don't mutate, because another kind of functionality of having that kind of bistable populations is, what if you want a repeatable treatment? And also you mentioned things about burden. So let's say you can keep it at the quiescent stage, right, at the small population level for some certain amount of time, hopefully at lower burdens, because it's not actually expelling any treatment, and then it's only expands and active when necessary. So this kind of structure, it has different uses. So even if the mutational robustness isn't necessary, the idea of it being bistable could potentially, if the application calls for, you could have some type of repeatable treatments because you're expanding and contracting back. And it could potentially be maybe less burden because in the lower level kind of quiescent state, you're not actually, basically making the toxin or making the thing that's necessary to treat the infection.
I see, that completely makes sense. So thank you. So that's amazing. I see now 11:04, so we probably need to close and then we continue to discuss. Okay, let me close. So thank you all for joining and staying today. We'll meet again next week on September 14, Thursday, the same time, the same Zoom link. And we'll have the Professor Pam Silver at Harvard Medical School and Dr. Seung-Hwan Lee at MIT. As usual, the follow informal chat will occur without recording. So please stay here if you are interested in chatting with today's speakers, and I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Thank you. Just one second.